collaborative and innovative environment will be our catalyst for the discovery that will shape the coming years and beyond. As we celebrate our successes, let us learn from the challenges and embark on the next chapter of our journey with a renewed enthusiasm and determination. Let us also recommit ourselves to the pursuit of excellence, innovation, and the advancement of knowledge for the betterment of the society. I would like to appreciate all the brilliant minds that have been part of the community since inception. The founders, the community managers, volunteers, scientists, researchers, educators, enthusiasts that have been supporting and dedicated their time to the noble pursuit of understanding the world around us. Your contributions, no matter how small, has played a crucial role in building this community and has moved us thus far. And I am confident that together we will continue to achieve great things in the years to come. Special thanks to the anniversary community for their relentless efforts in making today a reality. And to all members, past and present, thank you for being the driving force behind our community success. To our guests, thank you for joining us in celebrating this special occasion. We cannot wait to learn from your wealth of knowledge. Together, let us continue to explore, discover, and inspire. Once again, welcome to our 3rd anniversary celebration. Thank you for being a part of this special occasion, and I hope that you learn from it and you can also grow and thrive in your chosen field. Thank you.
Thank you very much, Esther, for that beautiful. Can we hear me, please?
Yes, we can. Yes, we can hear you.
So sorry, I joined quite late. I was having some technical issue. Hello, everyone, once again. My name is Idris, and I'll be moderating today's event. I know we've practically been welcome to our 10th anniversary, and I'll be co-hosting today's the first day with my co-presenter. So my co-moderator for today is Rukoya. Hello, Rukoya. Are you on this call?
Yeah, I am. Good evening, everyone. Welcome to tonight's session.
Yeah, thank everyone that have joined so far. You're welcome to the first day, and I believe Esther will give you a brief intro as to welcome us to the 3rd anniversary of Science Cafe with the theme Bioscience in a Changing World, Setting the Pace for the Next Generation. I know we're all excited and we can't wait to kick off the program. We already know it's a 2 days program, and today is the one. Follow up with the other subsequent tomorrow. Today we'll be having 2 speakers. Just before I introduce our first speaker for today, I would like to set some ground rules. Please, all participants, let's ensure that our mics are muted. If you have any questions or comments, we can use the chat box. Also, during the question and answer segment, you can use the hand emoji, and we'll take as much questions as we can during those segments. Please, our speakers will have 30 minutes for their presentation, and thereby we'll have 15 minutes for questions and answer segments. Our first speaker for today is Dr. Jaiyesimi Olakunle. He is a visiting postdoctoral fellow at Harvard University, where he is deploying untargeted metabolomics to eliminate genetic mechanisms controlling development using the Drosophila model. Dr. Olakunle has his PhD in natural and synthetic products / metabolomics from the University of Sao Paulo in Brazil. And tonight, as our first speaker for this 3rd year anniversary, he will be discussing with us Bioscience today, what you need to know. For those of us who were privileged to attend the last year, you could remember that one of our first speakers discussed with us Bioscience and entrepreneurship. And this year is a top notch. I'll be having Bioscience today, what you need to know to kickstart our talk. Please, Doc, you have 30 minutes for your presentation, and thereafter we'll have a question and answer segment. Please, do I have our guest presenter, Dr. Jaiyesimi Olakunle on the call, sir.
Yeah, thank you very much. Can I share my screen?
Yes, please, you can. You have the floor.
Okay. Okay, so yeah, so can you see my screen?
Yes, we can.
Okay, thank you very much. First and foremost, let me appreciate Science Cafe for inviting me to present at this occasion. And let me also congratulate Science Cafe for, marking or celebrating your third anniversary. The third year, I mean, every 3 years, quite significant. 3 is a significant number. For me, personally, 3 is also a very significant number. And, we'll be talking about all of that later in future. So congratulations. I hope that you walk into greater things as you move on, wonderful things you're doing. So today I will be presenting on this. So my name, like I said, is Olakunle Jaiyesimi, and I'm a postdoc at Harvard. So today I'll be presenting on this topic, Bioscience today. Can you hear me okay?
We can hear you well and okay, loud and clear.
So I'll be presenting Bioscience today, what you need to know. You will agree with me that this topic is quite vast. Bioscience itself is very elaborate. And then what you need to know, is vague and very broad, because one, I do not specifically know all the individuals that are listening to me today to be able to gauge what they need, what you need, So what I've just tried to do is to present something that I think you may need to know, and then you'll be able to connect with it based on your preferences, your individualism, your personalities, your interests, your experiences, background, likes and dislikes, and all of that. What you want to achieve in life, you'll be able to connect it with, whatever it is I'm presenting. It would have been better or lovely if I had known every person individually and I can be able to figure out, what to present, because I would know what you need to know. So the outline basically is, a brief definition and a brief history and evolution of biosciences, key areas of bioscience, introduction to metabolomic studies. So metabolomics is what I use, and it's one small aspect of bioscience, and so I'm just going to present more of metabolomic studies, and then talk about the opportunities, applications, future directions, and ethical considerations. So to definition, so we have bioscience, and of course, that's the science of biological systems. So it's good for us to be able to define what that is first. And so science, of course, is the knowledge of any kind, so I checked Google, thinking that, we must agree on what bioscience means. So if you go to Google and just type science, what does it mean? This is what it's going to bring out. So the systematic study of the structure and behavior of the physical and natural world through observation, experimentation, and the testing of theories against the evidence of things. So essentially, bioscience is the study or the knowledge of biosystems, biological systems, living systems, and all of that. So what do you need to know about the knowledge of biosystems or the study of biosystems, and what opportunities abound there? So that should form the crust of what I'm talking about today. So what is bioscience? Sorry, this Zoom. So what is bioscience? I'm sure that, of course, everybody can pick this up, wherever, on ChatGPT, on Google, in your textbooks and all that, but so it's a branch of science that applies biological principles to study living organisms and interactions with each other and their environment. And it covers a wide range of fields, including biotechnology, genetics, molecular biology, biochemistry, and variations of this. I'm sure you'll remember basic biology, where we talk about phenotype being equal to your genotype and your environment. So your genotype and your environment playing very significant roles in specifying, your phenotype. So you have the latent genes interacting with themselves and of course with, the environment to determine what happens to you. So that's what bioscience tries to focus on. What happens to this individual, and also another perspective I would like to share is that science is the study of what exists, within the biological system, what had been created, what subsists. So science is trying to unravel these things that exist, So we probably might not have discovered them. Some things are, buried in the ground or in hidden knowledge. We don't know about them yet, but they exist. Science now finds out that this thing exists, gives it a name, tries to understand it well, using all ramifications. For me, I would use metabolomics, to unravel what's, whys, and hows about that new discovery. And so that's what bioscience essentially does. And there are different fields, within this same field, this same principle where you are trying to uncover, what already exists. How does it exist? Why does it exist? What does it do to exist? So there are different fields, and that's where you have biotechnology, genetics, molecular, but all of these are just, different arms of the same organism, And so what bioscience does essentially is to probe the mysteries of life, understanding how living things work, and apply this knowledge to solve real-world problems in health, environment, industry, or what have you. Sorry, I'm needing to be moving the figures around. That's, the videos. So, so I'm going to go on to the brief history and evolution of biosciences. So bioscience, the history of bioscience itself is a fascinating journey through time. And it's marked by groundbreaking discoveries and innovations that have fundamentally shaped our understanding of life. And, oh, wow. Okay. And in ancient civilizations, of course, pre-500 BC, early humans intuitively understood biology primarily for survival needs, identifying edible plants and understanding animal behavior. And of course, this practice is still ongoing amongst us, where you sense things. Without systematically studying them, you sense them, you observe them, and then you generate hypotheses for which you're going to now study and establish if it's valid or not. And so ancient civilizations like Egyptians, the Greeks, began systematic biological studies, with philosophers like Aristotle making significant contributions to biological knowledge. And in the Middle Ages, the progress in biological sciences was relatively slow, and scholars like Avicenna made notable contributions in the Islamic world, particularly in medicine and physiology. And then from the Renaissance and early modern period, the invention of the microscope revolutionized biology, allowing scientists to observe microorganisms for the first time, and significant progress were also made in anatomy and physiology. And then in the 19th century, it marked the turning point in bioscience, highlighted by Charles Darwin's theory of evolution through natural selection. And then advances in cell theory, which is widely practiced today or studied today, established that all living things are composed of cells, the basic unit of life. And this also constitutes one of the mysteries of life. From one single cell, you have all the diversities that you can imagine, even within the same species. And then in the 20th century, biological sciences grew exponentially, and the discovery of the structure of DNA was a monumental milestone, which lays the foundation for modern genetics. And then rapid technological advances such as electron microscopy, molecular biology techniques led to significant discoveries in cellular and molecular biology. And then now in the 21st century, we've witnessed the rise of biotechnology, genetic engineering, including the completion of the Human Genome Project, which catalogs the human genome. And bioscience has become integral in addressing global challenges like climate change, disease outbreaks, and sustainable development. It's a big relevant, and there are different parts of it that you can hold. So throughout history, bioscience has evolved from a basic understanding of natural phenomena to a sophisticated and diverse field, deeply integrated into many aspects of modern life, from healthcare and agri to environmental conservation. So I go on to key areas of bioscience. So you have biotechnology, which involves the use of living organisms and their components to develop new products and technologies for various industries, including healthcare and agriculture. And then you have genetics, which includes the study of genes, hereditary, and variations in organisms, providing insights into inherited traits, evolution, and genetic disorders. And molecular biology, which examines the structure, function, and interactions of biological molecules, such as DNA, RNA, proteins, to understand cellular processes, and especially also focusing on what they do. And then biochemistry, which is what the preceding field like molecular biology does. Biochemistry involves investigating the chemical processes and substances that occur within living organisms, studying the molecular mechanisms behind biological functions or phenotypes or whatever it is you want to study. And so I go on to introduction to metabolomic studies. So first, when we consider the central dogma of life on the backdrop of bioscience, remember I said bioscience involves all this I've talked about. But when you consider the central dogma of life, which considers the fact that the genome produces the transcriptome, produces the proteome, produces the metabolome, which produces the phenotype. Can you see my cursor? Let me see.
Yes.
Okay, I will just use this. Okay, so considering the central dogma of life, you have the genome, which is what can happen because sometimes it's latent. I mean, nothing is happening, although it's unique in every individual. But when this genome is transcribed, when it's expressed, when the genes are expressed, you have the transcriptome or you have the transcript, the RNAs, and then when these RNAs are translated, you have the proteins, which may constitute enzymes, that may produce the metabolome, which are the small molecules, the metabolites. And these metabolites are the most proximal to the phenotype. So the metabolites or the small molecules or the metabolome actually specifies the classes of phenotypes that you may have. So that's one of the reasons why the metabolome is very, very important, and that's the area where I study. And so, so metabolomics is the molecular measurement of phenotype, all the characteristics that result from the genotype's interaction with the environment. And that's what this is just showing. So why are metabolites or small molecules? Remember, DNA, RNA, proteins are all metabolites, but I'm talking with respect to small molecules. So when we talk about metabolomics, we're talking with respect to small molecules, and I will talk about that as we go on. So why are metabolites, small molecules relevant? Because they are the canaries of the genome. They sing the song of the genome. So the genome encodes a lot of music, a lot of songs, a lot of notes, and all of that, but they cannot sing, but the metabolites are now like the canaries of the genome. So metabolomics is an emerging field, and it studies the global collection of an organism's small molecules. Those are small molecules that have less than 1,005 daltons. And then it takes a snapshot of an organism's metabolism. So by time of sampling, if you carry out metabolomic studies, it's going to tell you exactly what metabolic state the organism from which you had sampled was experiencing at the point of sampling. So it takes a snapshot of an organism metabolism, and then you can use that for your biological interpretations as you want. And so metabolomics essentially is a comprehensive measurement of all low molecular weight molecules in a biological specimen. And so what can, I'm sorry, I'm moving a lot of things around here so I can see. So what can metabolomics measure? It can measure carbohydrates. Most of the small molecules, all of the small molecules, carbohydrates, lipids, amino acids, vitamins, secondary metabolites, and metabolites generally involved in specific pathways, such as the glycolytic pathway and Krebs cycles. And of course, modifications of all of these small molecules can also be measured by metabolomics. And what can metabolomics not measure? What metabolomics cannot measure include genes, proteins, pathogens, and cellular structures such as mitochondria and the nucleus. But mind you, metabolomics can be used to measure the products of these items. So what the genes are producing, metabolomics can measure. What the proteins are doing, what they are producing, metabolomics can measure. Pathogens too, there are unique signatures. There are signatures of these pathogens sometimes. And so you can use metabolomics to actually, determine or identify the biomarkers of these pathogens and be able to determine what possibly pathogens, what bacteria, what viruses are within your biological system using metabolomics. And of course, you can also use metabolomics to study cellular structures, but not measure them as a web, but measure their products. So why can't metabolomics measure this? Because they are too large, to be detected using metabolomics methods. So overall, metabolomics is a powerful tool that is used to measure a wide range of small molecules, and it is valuable for researchers who study the way the body works and for clinicians who are trying to diagnose and treat diseases. And so what are the limitations of metabolomics? First off, I need to mention that there are two types of metabolomics. So you have targeted and untargeted metabolomics. Usually we say that untargeted metabolomics is global, is comprehensive, is unbiased, but I should tell you ahead that there is nothing that is unbiased. There is nothing that is comprehensive. Even metabolomics is not global. It can be global, it can be untargeted, but it is not unbiased. I just said to mention that. And let me see. Okay, so it can be difficult to identify all of the metabolites in a sample. So these are areas of improvement. These are areas you can come in with your own skills and improve. And if you can solve some of these challenges, of course, you are creating a lot of opportunities for yourself. So metabolomics is a complex and expensive technique that requires specialized equipment and personnel. And the levels of metabolites in the body can be affected by a variety of factors, including diet, exercise, and stress. And so this can make it difficult to interpret metabolomics data. So, what you just do is to keep all of these variables constant, while designing a metabolomics experiment. And metabolites can be unstable and can degrade over time. So you factor this in. How do you store your samples and all of that? So you can get away with it. But yet, metabolomics is a powerful tool that is useful for understanding organisms' metabolism and for diagnosis of diseases and monitoring of disease prognosis. Again, like I said, small molecules are the most proximal to phenotype. So by being able to measure these small molecules, you'll be able to classify, your phenotype accordingly, even more appropriately. But now metabolomics becomes even more powerful when you are able to integrate it with earlier tools, by that I mean proteomics, transcriptomics, or genomics. And then you have, almost a very comprehensive view of the biological system. And so information that you can derive from small molecules include the metabolite classes. Of course, with the collection of small molecules you have been able to measure, you can assess what metabolite classes predominate within the biological system under study. And then, of course, the enzymes or the proteins that, work in concert or in a network to produce these small molecules. So using small molecules alone, using metabolomics alone, you can have an idea of what enzymes or proteins are involved. So imagine integrating metabolomics with proteomics. It gives you exactly what is actually going on at the level of small molecule biosynthesis. But even without proteomics, small molecules alone can still give you an idea of what enzymes are involved, in their biosynthesis. And of course, metabolic pathways. So metabolic pathways consists of small molecules. So with collections of small molecules, you can run pathway analysis that can tell you which pathways are enriched, versus the other within your biological system under study. And then, of course, the DNA and mRNA that produce the codes for the enzymes that produce or biosynthesize these small molecules can also be determined using metabolomics. And so the phenotype ultimately. So I'll just leave it at that. And then the application of metabolomics include drug discovery. So metabolomics enables the identification of metabolites, including natural products from natural sources, plants, marine animals, and all of that are bioactive with potential as leads for drug discovery. So this applications also gives you an idea of which areas you can apply metabolomics, which areas there are career opportunities for those who are pursuing metabolomics. And of course, in concert with probably other omics tools. But so I'm working and presenting on metabolomics in this area. So drug discovery and personalized, I can see that, personalized nutrition by using metabolomics to analyze an individual's metabolism. Personalized nutrition plans can be created. Indeed, personalized medicine also. So by using metabolomics to study the reaction of an individual to certain drugs, what's the outcome, of certain treatment strategies, you can personalize the treatment for such individuals. And then precision medicine, where metabolomics can identify biomarkers that indicate which treatment will be most effective for an individual, and then you can make informed decisions based on the application of metabolomics in healthcare. And then environmental metabolomics, where you can use metabolomics to detect the metabolic responses of organisms to environmental stressors, such as pollution and climate change. Food metabolomics identifies metabolites that are present in foods to assess their nutritional value and quality. And finally, here, clinical metabolomics, where you use metabolomics to measure metabolites as biomarkers for diagnosis and monitoring of disease progression and treatment. And so eventually, sometimes if you integrate metabolomics with phenomics and transcriptomics, you can pinpoint certain pathways or certain genes, that may need to be optimized, perturbed to optimize a particular character or phenotype or behavior, And so this is a previous research that was done. And essentially, I don't want to go through the details fully, but, what was done essentially was looking at how bacteria, a particular bacteria that is associated with cystic fibrosis, responds in a routine laboratory media, a laboratory medium that had been used regularly for the study of this bacteria, versus how the bacteria responds in a synthetic cystic fibrosis sputum medium. And that's a medium that mimics, the cystic fibrosis lung environment. So I used metabolomics to try to determine what happens with this Burkholderia cynocepacia in these two different environments. How do they respond? And of course, you can bet. So, this is just the pipeline, and LCMS was used, and then the data was extracted and submitted to different platforms. And so looking at these two, there are 3 species. And so you see that in one, let me move this away. So you see that in one condition, you have almost 900 unique metabolites. In another condition, you have almost about 600 unique metabolites, of course, in the presence and absence of trimethoprim, And so at the end of the analysis, we found that in the presence of trimethoprim, Burkholderia cynocepacia K56.2 biosynthesizes, in the presence of trimethoprim, biosynthesizes 362 unique metabolites compared with 59 metabolites in routine laboratory medium. So using metabolomics was able to point out that simply using this laboratory medium to study Burkholderia cynocepacia, we may be losing significant information that we're supposed to actually have, if we use synthetic cystic fibrosis sputum medium, which mimics the cystic fibrosis lung environment, and then we can actually make discoveries and be able to provide, adequate information to healthcare professionals, who are treating or managing cystic fibrosis in their patients. And so the value of learning metabolomics includes developing key skills. So learning metabolomics techniques can improve your analytical and problem-solving skills, making you a more valuable asset to any research team. Explore exciting career opportunities and options or whatever. There's a problem with that. Okay, explore exciting career options. So metabolomics is a rapidly growing field with a wide range of career opportunities, including academia, biotech, pharmaceuticals, and healthcare. You have a whole lot to choose from. So there's no lack of career opportunities for you, because even the field is still growing and we are still getting to understand what potentials are available here. And so you make breakthroughs and discoveries, even in your personal life, at work, wherever, whatever it is you're applying metabolomics to, because metabolomics has the potential to drive groundbreaking discoveries in many fields, including neuroscience and precision medicine. But these are just, two that I chose to mention, So let me see, I'm having trouble moving all these things around. So finally, conclusions and key takeaways. 1, metabolomics is a powerful tool for studying the small molecules that make up the building blocks of life. What's the tool? It has a wide range of applications, including drug discovery, personalized nutrition, and precision medicine. And learning metabolomics can develop your skills, explore exciting career options, and open up new avenues of research. And finally, as part of the things I talked about, metabolomics can help us understand the molecular basis of behavior and the complex interactions between genotype and environment. For the sake of time, I've tried to condense most of these things I talk about, but it's actually, they're all actually pregnant. And so finally, the ethical considerations, scientific responsibility is very key. So researchers in biosciences, so while I'm talking about biosciences, you are thinking about metabolomics, because for me, I need to be responsible scientifically within the field of metabolomics. So whether you are in genomics, you are in transcriptomics, you are in proteomics or metabolomics or any related field, these ethical considerations apply. And so researchers in biosciences have a responsibility to ensure that their work adheres to ethical guidelines, promoting the well-being of humans, animals, and the environment. Gene editing controversy, of course, so at the end of all of this analysis, molecular analysis, whether you're using metabolomics, transcriptomics, proteomics, or genomics, I think the hallmark is their ability to be able to change the fate of the biological system, especially to be able to improve it. If there are certain genes that are contributing to a disease state, or certain genes are contributing to maybe the taste of certain fruits and that are not desirable, and you want to change that, of course, you can apply all of these techniques within genomics, transcriptomics. However, there are controversies that are associated with them. And so the advent of gene editing technologies like CRISPR, Cas9 has sparked debates in ethical implications, such as designer babies and potential misuse. So if you are working into biosciences, you also must be aware of these controversies. And what's your opinion about it? What do you think about it? How will you approach it if you were given the power to make a decision about gene editing practices? And so privacy and genetic data, the collection and analysis of genetic data raise concerns about privacy and the potential misuse of personal information. And then the exciting journey ahead of metabolomics, of biosciences, essentially. The field of biosciences continues to evolve rapidly, driven by curiosity, innovation, and the quest to unravel the mysteries of life. And you can be sure that the future holds a lot of promise, especially with the advent of artificial intelligence. So imagine integrating artificial intelligence with biosciences. Imagine what can happen. So this is a good time to be alive. This is a time for, most people, I encourage everyone to join the fray of biosciences. Also because our purpose on Earth, is based on, what do you call this? Based on these, I can't remember now. If I remember, I will mention it. But, subject, predicate, object. So biosciences is based entirely on that. Subject, predicate, semantic triples. So it's based on semantic triples. And semantic triples talks about subject, predicate, object. So beyond biosciences, even in your personal life, we are all doing something related to semantic triples. Subject, predicate, object. Today, Olakunle is presenting a talk. That is semantic triples. If I was sleeping at this time, Olakunle is sleeping. That's semantic triple. So in biosciences, essentially, the genes produce the transcripts. The transcripts produce the enzymes. The enzymes produce the small molecules. The small molecules specify the phenotype. That's semantic triple, and that's my talk for today. Thank you very much.
Thank you so much, Doc, for the wonderful presentation. It's been an interesting ride. At some point, I couldn't even catch up with my jottings. I hope you'll be able to share your slides with us after now, sir.
Yes, I'm happy to do that. I'm sorry I was too fast.
We understand the timing too may not have helped in that regard, because also we have another speaker later on. Thank you very much, sir, for the presentation.
Absolutely.
It was an eye-opener for me as a biochemist. It showed me some of the beauties of metabolite at its small levels, even in how essential it can be, both at the environmental level and in understanding disease and nutrition. Thank you wonderfully for the lecture.
Thank you.
I know several of us or several participants will have a question. We can choose to use the chat box and put our questions, or we can use the raise your hand emoji, and I'll pick as much as we can. Though we are lost for time, but we'll try to answer as much questions as we can. I think while our presenter was rounding up, I saw a question in the chat group. This one is from Saifullah Mustafa. He asked, how can metabolomics be used in food science to know the nutritional content of a particular food?
Okay, that's a very low-hanging fruit, because you shouldn't limit yourself to, I understand this question, but, you should even think beyond it, because metabolomics will definitely do this for you as a low-hanging fruit. That's what metabolomics is meant for. So if I'm to answer the question, you collect your food, you extract it appropriately. There are different extraction protocols that you can use depending on the kind of food and what you have, what you intend to achieve with, the profiling. So you can then subject your extracts to LCMS, GCMS, mild DMS, and all of that. So there are different, ways of doing it, but it is very possible. But beyond that, for me, the question would be, what's unique in this food and why is it there? What's it doing, in terms of health implications? But metabolomics can definitely be used for to determine the nutritional content of a particular food. If you have any questions or you need any help with regards to that, you can always get in touch with me.
Thanks so much, sir. Another question is, I think when you were trying to explain about some of the research you carried out in the last paper you shared, someone asked, were you able to easily isolate new or promising bioactive metabolites?
Not in that paper, because the objective of that research was just to understand what is different. And interestingly, yes, because you see, we gave trimethoprim, to the bacteria. So we expose the bacteria to trimethoprim, and trimethoprim is one antibiotic that, doctors use to manage cystic fibrosis. And what we found using molecular networking on GMPS, that's a kind of analysis, we found that trimethoprim was connected to certain motifs. So certain structures that shows the modification of trimethoprim by bacteria, which means, or which suggested that the bacteria in the cystic fibrosis sputum medium was modifying trimethoprim in such a way that trimethoprim will lose its effect. Those were the new compounds that we found, but not bioactive metabolites as it were, because of the nature of the research or the research design. But of course, metabolomics has actually exposed during my PhD, exposed or unraveled certain new metabolites, which were promising as new bioactive metabolites that were isolated, and then I characterized them using NMR, and they were new, so using molecular networking. So I'm throwing that word out so that you can look it up if you are not familiar with it. So metabolomics, if you use molecular networking, it connects your molecule of interest to similar other molecules that may not have been identified previously, and so not have the identities are not available in databases. Also, they may be new, but they have similar structure with a known structure. So with that, you can propagate your annotation and then identify new compounds that can be isolated and tested.
Yeah, so in a case, I want to quickly ask this one on a personal level. In a case where you had more compounds compared to the other media, would you say suggestive of? What would you say suggestive of?
If I had more compounds in the routine laboratory medium,
yes, compared to the normal one.
Yes. Now, whatever, the research design was to show, what actually happens in the biological system, in the real world situation, in the cystic fibrosis lung. We wanted to know what happened. So if we had extremely more compounds in the routine laboratory medium than the cystic fibrosis medium, what is just saying is that, what we are seeing in the lab with the routine laboratory medium is not representative of what actually exists in the natural world in the cystic fibrosis lung. Because in the cystic fibrosis lung, you have only 10 metabolites that are being biosynthesized when the bacteria and the lungs are interacting. But in the lab, we are seeing 1,000,001 compounds. So something is wrong. And I will tell you that one of such challenges may be maybe from trypsin or certain proteins that are in the routine laboratory medium. During digestion, most of those metabolites get digested. So if you have more in the routine laboratory medium, it's coming from somewhere and you can trace it back, to where it's coming from. What is different between this routine laboratory medium and then this cystic fibrosis lung synthetic medium? What's different and where are these things coming from? And metabolomics will help you to be able to identify the compounds and be able to know that, no, this is not supposed to come from this natural medium. It's not coming from natural medium. And so where is it coming from? You can trace it back. It's easy to trace back, because you know what you have added into the routine laboratory medium, which can be degraded, broken down to produce these small molecules. So you know where to go.
Thank you very much, Doc, for that answer. Thank you. Shakir Adeyinka is asking now, please, what advice would you give an undergrad or early career researcher that wants to build a career in metabolomics? What do you think they could start from?
Now, the advice I will give you is to pursue it. But let me say this, and I gave a presentation some time ago, and I said the same thing. I am here today using metabolomics. I did not, and I say again, I did not actively choose to do metabolomics. But today I look back over my life and I say, what would have happened if I never chose metabolomics? Or if metabolomics never chose me? So metabolomics chose me. Why did metabolomics choose me? It chose me because I followed my passion. I followed my interest, my questions. I had questions I'd been wanting to ask. So while I was in Nigeria at Obafemi Awolowo University, Ile-Ife, I was in pharmacognosy. I was a pharmacist student, but I did my PhD, I did my, project in pharmacognosy, which is medicinal plants, because I was interested in understanding how medicinal plants work. Now, my master's I did in pharmacognosy again, and I only fractionated, the extract, so I got different fractions, but I did not know what small molecules were active in it. So I was still very much interested in knowing what compounds were active. So I applied for a PhD in Brazil, And I did not talk about metabolomics at all, and then the supervisor, the prospective supervisor at the time, read my proposal and said, okay, it's good, but please, can you include metabolomics? Because he was reading what I wrote and he found that metabolomics will answer most of this question. And that was how I got into metabolomics. So I learned metabolomics during my PhD and I had to do it because my PI at the time, thought it was good. And I am so grateful. I would say Olodumare, I believe that Olodumare guides the steps of people, and I have out, with regards to that fact that, he probably must have guided my steps to it, whether you believe in that or not, you must believe in something. You must believe in yourself or in your God or in your Eleda or, whatever it is to guide your steps, but I think that most importantly, you have your questions that you are interested in, have your questions that you're interested in, continue to ask these questions with every opportunity you have, and the right paths, the right steps to take, will yield themselves to you, and so your career will choose you. That's the best I can say. Where you are currently is the most important. Where you are currently is the most important. Make the best of it. For me, Olakunle, the most important part of my life right now is talking with you guys. So this moment is the most important. I want to make the best use of it. Because if I make the best use of it by letting you know who I am, my passions, my interests, and all of that, what I need for my life will open up. And the same thing with you, whatever schools you are, whatever it is you are doing, whoever you are meeting, just make sure you have your questions on your fingertips, on your lips, your passions, your interests, your questions should be all about you. And always let people around you, immediately around you, know what you want, what you are interested in, what you're passionate about. And I think that the universe will yield itself to you.
Thank you so much, Doc, for that answer. We will not be able to take much, but we'll take more questions, and after which we would proceed to the next segment. But meanwhile, the next question is: where do we classify lipidomics in the omic research landscape?
It's metabolomics, because lipids are small molecules.
That's true. Okay, Doc, thank you very much. We would love if you could share us your slides and as well as give us your email for any further contacts that some persons are. Thank you. We'll be wonderfully wondered. I don't know if there's any other questions. I'm still going through the chat. Are there any other questions? Is there anybody who wants to use the mic? You can please signify by raising your hand. Okay, this one goes thus. Considering this importance of metabolomics that you have talked about in bioscience, what is the state of this field in Nigeria, and what procedures are needed to be able to advance the field in Nigeria? What advice can you give for upcoming scientists in Nigeria who are interested in this field?
Yeah, the state in Nigeria is not really advanced because of very obvious reasons: no electricity, a lot of challenges, the instruments needed are not there, and all of that. But I mean, there are ways to go about it. I don't think that there should be limitations. And as such, for me, I'm personally thinking about, I've started working on it, started thinking about how to transfer knowledge, how to transfer this technology to Africa. So I'm creating a platform, where whether you are in Nigeria or in a village somewhere in Africa, you will still have access to these cutting edge technologies around the world, So it's something I'm still working with. So even on the 12th, December 12th, I'm meeting somebody who may be able to develop a virtual platform for instrumentations, such that wherever you are, you can still access it. So it's a whole lot of things going on in the background. So yes, it's not so good yet, but I think that it's possible. And everybody needs, I love this question because, you are thinking ahead. Everybody should be concerned. How do we globalize? How do we make things happen in spite of the challenges, in spite of our leaders? How do we make things happen? The most important things, the things that should happen, the right things. So, the fingers crossed.
Okay, Mutiat, I noticed you raised your hand. Do you still want to ask a question?
Yes,
Mutiat,
it's
are you please
Moina.
call? Okay, please.
Okay, yeah, I am.
Okay, please
Thank
go
you
on.
for the wonderful presentation, sir. I know
Cool.
it's quite a challenge to discuss bioscience because it's such a wide field. So my question is, so I've discovered that there is a particular type of omics that we have now, interactomics. Just said about it. I think it was discovered in the 90s, 1995 to be precise. So can we classify all the omics under interactomics, like interaction, so omics? That's my question. Thank you.
Yes, you can, but at the same time, I would think that interactomics focuses more on proteomics. So proteomics is like integrated, it's like the fulcrum. It's the fulcrum that connects everything together. So it connects genomics, transcriptomics with metabolomics, So so if you have some transcripts that are not producing proteins, they can serve as transcription factors and all of that. So at the end, they're regulatory. So at the end, you have more or less of transcripts, And so if there are no transcripts that are protein coding, you're not going to have proteins. If you don't have proteins, you're not going to have small molecules. So the proteins forms like the basis of almost everything. So I think interactomics, focuses more on that, on proteins. But yes, as you would think, interactomics talks about the network. So today, people we use different terms to try to explain different things. Some people today are talking about MGUAS, where you have metabolomics-based or metabolome-based genome-wide association studies. So today you have integration of all of these omics into multi-omics, where you can have very comprehensive view of the biological system. So essentially, that's what I think it is.
Thank you very much, Doc. Thank you for the time, for honoring our invite to this 10th anniversary. We are so much grateful. I know a lot of persons have more questions, and I'm seeing some questions in the group too, but because of time, I will not be able to take any further questions so that we can go to the next segment. But please, I know a lot of us will be reaching out, sir. So we'll be interested in the slide as well as your email, please.
No, you can speak with Emmanuel.
Okay, we'll do that.
Slides and all that. Yes. Thank you very much.
Thank you, sir. Thank you for having us. Thank you. Okay, guys, I believe we enjoyed that session. It's been a beautiful one. Thank you for the questions. To those of you who could not take your questions, we're so sorry because of time. We're very, very sorry. Please pardon our excesses. Now we'll quickly want to go to the next segment of today. Remember, it's a 2-day affair, and today is the first day of the 10th anniversary. And so far, we've had a talk on bioscience today, what you need to know. A lot of us have been introduced into the world of metabolomics, and why I for one, am showing a keen interest in this. So permit me to invite my co-moderator for today, the person of Rukaya, who will be taking over for the next session. Over to you, Rukaya.
Okay. So yeah, thank you very much, Idris, for the wonderful moderation so far. And thanks to our first guest speaker for tonight. So once again, good evening, everyone. My name is Rukoya, and I will be moderating the second segment for tonight's event. Still standing on the existing protocol that all participants should stay muted to avoid distraction. And thank you to everyone so far for maintaining decorum. So yeah, our second guest speaker for tonight is from Bamgboye, Adeboye. He's a PhD student at the University of Minnesota. So Bamgboye obtained his pharmacy of bachelor degree from the University of Ibadan. His undergraduate research focused on the anti-convulsant property of lemongrass using animal models. So following his graduation, he undertook a professional internship in academia, working on toxicology and neuropharmacology projects. He also has extensive experience in pharmacy practice and health insurance. So now he's currently pursuing graduate study in clinical pharmacology and Biostatistics at the University of Minnesota under Dr. Lisa Cole, with a primary interest in applying clinical pharmacology, pharmacostatistical, and machine learning method to optimize the treatment of seizure emergency and prevent post-traumatic epilepsy. So yeah, and our guest speaker will take us through the topic technological advancements in bioscience research and introduction to how. I'm sure we already have the R application downloaded and installed on our device, and we are ready to learn from the wealth of knowledge of our guest speaker tonight. And I hope our guest speaker is ready for us and is ready to take us through the technical event. So after his presentation, the question, answer, and comment section will come after for 15 minutes. And during the presentation, if you have any question, you can use the emoji to raise your hand, or you can put your question in the chat box. So Bamgboye,
yes,
I hope you are ready for us. Yeah.
I'm here.
Yeah. So yeah,
First
please
of
be
all, can you
reminded.
hear me?
Yeah, we can hear you. Yeah.
Like very well.
Yeah.
I'm using a different device to run this. So I just need to be sure that everything is working well. So just let me know if you can hear me clearly. Okay. Thank you so much. And good morning, good afternoon, good evening, depending on where you are. It's such a lovely day here in Minneapolis. It's like 9 degrees Celsius, but it's given the time of the year, it's it's warm. So I would be, okay, let me share my screen. Just give me a minute with that. Okay, so let me know if you can see my screen.
Yeah, sure, Claire, we can see your screen.
Okay. All right. Okay, so it's so nice to be here, and I would not like to waste any more time because I know we'd probably have questions down the line. So I'll just jump right into it. So I'll be discussing advances in biosciences, and I'll be looking at a gentle introduction to R. So, oh, sorry. Okay, so this is the content. We'll be just doing a brief overview of biosciences as a multidisciplinary field, which the previous speaker has dealt with extensively. We'll look at a few emerging bioscience careers, in-demand skills for bioscience researchers, and then we move straight to R. Okay, so as the previous speaker said, like there's a lot going on in biosciences. Like living organisms are very complex, and because of that, there's like it's impossible for one field alone to actually or one discipline alone to be able to understand all of the components of living organisms. And so it makes sense that you break it into parts that allow us to study different aspects of the organisms, as well as how the organisms also interact with their environment. So biosciences is like a very huge field. And at the center of it all is just trying to understand the living organism from the simple level up onto like, or considering the different layers of complexities that show up there. So now there's no uniform agreement for what constitutes bioscience or what field is actually bioscience, because these days you have new fields like biomedical engineering and all that getting into like if you have things like tissue engineering and like a couple of other fields like that that just tend to move into answering questions in the biological setting. Like you begin to understand how wide the field of biosciences is, and so there's no uniform agreement for whether or not this constitutes or this counts as a bioscience or not. But generally, if it has to answer a question involving living organisms, in a way, it still falls under the umbrella of biosciences. So generally, the idea is to start studying organisms from the very like basic level, like from the cells, and then you take that to the organism and then the ecosystem and as well the population. So there's quite a lot like going on in the field of biosciences. And so it's one of the most rapidly evolving fields, especially because we've also made significant strides in like technological advancement. And so most of those advancements have also been applicable to answering questions in like biosciences as a whole. And so we see that as technological advancements has continued to move forward, biosciences as a field has also continued to move forward, like in the same vein. So this just provides some idea of the disciplines that fall under the scope of biosciences. So you can see that it ranges from like plant sciences all the way to management. I mean, you might not necessarily consider that to be a bioscience, but if you think about how bioscience work now, or like how biosciences or bioscientists work nowadays, there's always some level of management involved in it. You might not necessarily count it as a bioscience, but a lot of times it's inherently part of how you advance the field of bioscience as a whole, especially when you think of things like grant application or management or recruitment or things like that. It's often a huge part of it too. So this just shows that there's really a wide range of disciplines that fall under biosciences. So of course, some of them are very like familiar, like botany, zoology, microbiology, immunology, but there are some of them that may not be too familiar, like bioethics or biodiversity or things like that. So it's just to show that there's such a wide range of things that could fall under bioscience. Now, for emerging careers in bioscience, there's a lot, and this is not exhaustive. The list here is not exhaustive in any way. It's just to highlight some of the ones that I found particularly interesting. So synthetic biologists, bioprinting is a big deal these days. Of course, there's tissue engineering, which is something that many biotech companies are looking into. There's precision medicine with advances in genomics and gene editing techniques, and understanding more about how genomics influence both response to therapies as well as predisposing individuals to certain types of medical conditions. Like that has really moved the field of precision medicine like forward. And so you have a lot of things that sort of spin out of that, like genomic counseling and things like that, which were not traditionally careers that people would consider themselves going into. But the fact that there are so many aspects of like, or will I describe it as like overlap between biological sciences and like medical sciences and advances in technology and all that together, there's just this we just tend to see that there's just a growth in the career opportunities that exist in this space. And one thing I would like to point out here is it's for many of these career like parts of these career opportunities, it's not, many of them are not traditional. You don't just end up in those fields because you went to study those particular fields. Most of the times you find out that you have some basic understanding of like any of the fields in biological sciences. You might have like a degree in immunology or something, and then you apply that knowledge to answering a specific question, maybe in a related field. Like nowadays that we're looking at immunotherapy in cancer, you have maybe a solid understanding of like immunology as it were, and then you apply that knowledge in immunotherapeutics. And so that's sometimes how people find themselves in some of these fields. It's not like they just saw it online and be like, okay, I'll just apply to this program. It's just that you have a set of skills that are grounded in biosciences, and then you find an opportunity to apply those skills in other areas or to answer questions in other areas. And that just, you just find that that's a lucrative career, and then you decide to pursue it. Look at something like science communications officer. Of course, we know that people do science, but communicating science to people is also a very critical thing that needs to be done, especially if you are trying to get grants, you're trying to find money to do something, you're trying to influence public opinion about certain things. You might have a very good background on writing. You might have like maybe a little bit of knowledge about biological sciences, and then down the line, you can apply that in a role such as a science communications officer. So an excellent bioscientist, regardless of whatever field you are in, needs to have like a wide range of skills. Like you need to be quick on your feet in terms of critical thinking and analysis. How do you approach a particular problem? You need to be able to design like an experiment. If you need to answer a question, being able to design an experiment to answer that question is going to be a skill that you would always need. You would need to understand ethical concerns. For instance, like with gene editing nowadays, what are the concerns that could come up? There are some of those things that are grounded in like, or that have legal consequences, which you should also consider. So that's something you would also need to learn. What is it that I can do? What is it that I cannot do? If you're doing animal studies, what kind of environment should I subject my animals to, and things like that. Of course, there's also collaboration and teamwork, because as far as I know, biological sciences as a whole are perhaps some of the most collaborative fields that you would ever find. Like, it's very hard for you to see a typical bioscience paper with just one author, compared to fields like math or computer science, where it's probably, or physics, where you could find like a paper authored by just one person. In many biological sciences, that's not the case, because you just need expertise from people that's in different areas. So you need to be able to collaborate with people, like do projects with people. And of course, there's also data analysis and interpretation. So being able to come up with like the result of experiment, presenting that result in such a way that it's easy to interpret and understand and make informed decisions with, is also something that is very important, like these days. And I would say it's perhaps one of the most important skills that a bioscientist should have in the 21st century. So still on subject of data science and interpretation, like these are some of the software that are currently available for people to use in answering one question, visualizing data, like working with data and all that stuff. Obviously, you're not expected to know everything. It's just to provide information about the things that are out there and available for you to explore, and how these things are really what a lot of people that are working in data science and interpretation use in their day-to-day work. So we've also had, just like I said, with the advancement in technology, there has been significant advancement in many of the applications of biosciences. So these are just some of the advances that we've had. Organ on a chip, of course, CRISPR is perhaps one of the most popular, with the Nobel Prize in 2020 being awarded in this field. It has just been witnessing unprecedented growth, and we hope to see much of that like in the future. Of course, machine learning, biosimulations, the image is actually organs on a chip, just demonstrating how the liver and kidney are connected, and how that could also be useful in certain types of medical research. So these are some of the advancements we've had in bioscience, and we hope to see more of them moving forward. Okay, so switching gears now to R. What exactly is R? It's a programming language like Python, Java, C, C++, whatever you're used to. It's interactive, meaning that you run the code. When you run one line, you get a response line by line. In contrast to some other types of programming languages where you have to compile all of the codes first before you get a response. So R is interactive in that you get to see the result of what you do on one line. It runs the code line by line, essentially. It's open source, so I mean, it's free to use. You can easily go online, download things. You have lots of YouTube videos. The software itself is free to use, so that's also a very nice thing about it. It's growing faster. It's very popular, especially if you're going into academic research. R is something that is really nice to learn, and of course, it's adaptable to many types of operating systems like Windows and Linux and all that. Now, basically in R, there's a base R, and then there are packages. So the packages sort of extend what you can do with base R. So if I need to find a way to do something in R, and I want you can look at it as a set of tools that allow you to do something that you want to do in R. And the good thing about R packages is that anyone can actually come up with an R package. And so you could figure out that, okay, I want to come up with a package that helps people maybe convert their data from one form to another form easily. And then you can write the code of that and add it as a package to R, and someone else can use that for some of their work. So right now, there are over 10,000 R packages that just help people use or manipulate their data or use their data in such a way that it's meaningful for them. And the problem with that is because our packages can be contributed by anyone, basically, there's some sort of limitation with that, because you really can't, what's it called? Because like it's been contributed by different people, sometimes the existing packages in R, or like the existing base R, are not compatible with some of the packages. So you have to, it just makes life difficult sometimes, because there's no harmonized way of doing things. So that can introduce some sort of like difficulty with using R packages. So essentially, it just helps you use R better. That's basically what R packages do. And so usually once you install the packages once, you need to install them like again. It's just somewhere in your library that you can always call anytime you need them. So using this library function just helps you call the package from where you've stored it. So you think of it as a physical library where you have books on the shelf. So one shelf or one book on that shelf is a package that gives you instruction on how to use or do something. And then so when you need to call a package, it's like you're walking into the library and picking that book off the shelf to use it for something, to answer a question. Two major forms of ways that information, or two major ways information is stored in R are vectors as well as data frames. So think of vectors as list of data values. So as you can see down, Science Cafe journal clubs, there are so many, like we have a couple of journal clubs in R. Some of them include like cover topics in, sorry, that's a mistake. So some of them cover topics in immunology, biotechnology, pharmacology. So the list of those could be considered like a vector. So it's a list of data values. Now, data could also be numeric. It could be character. It doesn't have to be numbers. It could be words. It could be categories. It doesn't have to be a number alone. And then, of course, data frames are more like data sets. Like those ones are much larger in terms of what you could find there. So this just shows you what a vector could look like in R, so it doesn't look surprising. And so the C there just shows that you are sort of putting everything in a bag. So immunology, biotechnology, pharmacology are all journal clubs inside of a bag, which is Science Cafe Journal Club. So that's basically what that line of code means there. It's okay. So these are some of the popular R packages. So to import, to tidy data, to wrangle data. Now, data wrangling has so many things under it, but I tend to look at it as just a way to process your data. Like it's just making it easier to handle. That's basically what data wrangling is. And of course, there are much more technical definitions, but in a high-level summary of data wrangling is just processing data. And then so you can visualize data, you can program and all that. So these are just some of the popular R packages. Like I said, there are over 10,000. So this is by no means exhaustive. So RStudio is also something you are likely to come across a lot. It's simply an IDE, which is an integrated development environment. In simple terms, it makes your life easier when you're using R. That's essentially what RStudio does. And to use RStudio, you also need to run R, because RStudio is kind of like an environment that is built within R for you to use R. So of course, you can't use an interface for something that doesn't exist. So you usually have to install both R and RStudio together. So essentially, RStudio is a very versatile tool, allows you to import, visualize, explore, plot data, all the other things listed here. So you can basically use RStudio for all of these things. Now, this is the RStudio environment. You could see it as white or black, depending on whatever color, depending on what your preference is. Black is my preference. I don't know. Once you start using black, it's hard to go back to white, but just pick what works for you. There's no right or wrong way to use your environment. But there are just majorly 4 environments or 4 panels. The one at the top left is basically your source panel. So this is where you write your code. This is where you store your code. So if I'm doing a project, like a group project, I want to write a code and I want my colleague to be able to run that code. So for me to ensure that I can reproduce that code every now and then, I would write the code in this top left corner, which is the source panel. Now, below it, at the bottom left of it, is the console. So this console, you can also write code in the console, but the problem with writing code in the console is that it's not saved. So if you close your RStudio environment and you open it again, a lot of times you are not likely to still find your code there. So if you want to find the code that you've been writing for like the last 2 or 3 days, write it in the source panel. That makes it easy for you to retrieve it. Now, at the top right is the environment. This basically stores all of your objects, your vectors, your data. Everything is stored in this aspect. If you want to import your data, it's also this side of it. You go to import your data. And then when you want to visualize things like plots, or you want to ask for help in R, the bottom right is where you do all of that. So just as a summary, the top left is where you write your code for reproducibility. The bottom left is where you can write code there. It's easy for you to see if you made a mistake in writing your code. So if you make a mistake in your source code, like it's going to be displayed down here. That's where the software has an opportunity to yell at you. So in all the other places, you tell the software what to do, but this is kind of where the software tells you what to do. And so this is kind of just for fairness, this is where the software can yell at you or give you instructions. And then the top right is it's basically your environment where you store your data frames or your vectors or your data. And then the bottom right of it is where you visualize or you ask for help from R. That's very simple, but the idea. So, well, people say programmers prefer dark mode because like light attracts bugs, but now it's just a joke, of course. Like you could run into problems whether you use light or dark mode. That doesn't save you from running into problems. But personally, I think I like black. And believe me, once you start using black, it's hard to go back. So if you need to debug, let's say you do something wrong and the software starts yelling at you, what can I do about that? Well, there are a couple of ways you can figure out what is wrong. One way is Stack Overflow or online forums on the internet. You can post your question there, join those communities, ask for help, network and find better ways to do things. Of course, there's your old friend Google. You can always type it. How do I make a bar chart or a bar plot in R? You can always type that there and you see answers. Or how do I add color to my bar plot in R? You can always start like that and you would get answers or a couple of answers in R. Of course, there's your new friend too, ChatGPT. You can always type the question in ChatGPT and ChatGPT will give you an answer. And ChatGPT is very nice, especially when you're running into problem with R, because it's also likely going to give you examples of how to run the code, or like maybe templates for you to run the code with, which is also very nice, especially when you're starting out and you might run into problems. Or you can also ask your colleagues. Okay, so the 6 stages of debugging, that's really unnecessary. You can look at that. It's just a fun bit. Okay, so now off to RStudio. So just for the remainder of the time we have here, please let me know if you can see my RStudio. Can you see my RStudio? Can someone see my RStudio?
Yes, we can. Yes,
Okay,
we can.
okay.
Yeah.
All right. So now it can be intimidating if you look at it here, but essentially this is just the same thing I showed, that you have 4 panels there. You can actually adjust the size of the panels by moving things up or down. Okay, so it's, if you want to see more of your source panel, you can see more of it. If you want to see less of like that, you can move that. You can also move your plots to see more of it or less of it. So it essentially it's interactive. Now in this source panel here, you can store your file. So you have an opportunity to store your file, like store the name of your file. Sorry, was someone asking a question?
I think that particular tab is not being displayed.
Oh, I'm so sorry. I'm so sorry. Just let me. Sorry. Let me. Okay. So can you see it now? Okay, so it's loading. Okay. Yeah.
Yes.
I'm so sorry. I'm sorry about that. Okay, so you can just what I was saying, you can adjust the sizes of the panels to see what you need to see. If you want to see less or more of one, you can see that. So at this top left here is the source panel. So if you want to write a new line of what we call R scripts. R scripts are like a new document in R. So just like if you want to write a new Word document, you have to open a new Word document to write it. So in the same thing in R, but those ones are called R scripts. So if you want a new document that has your code for maybe a different project, this is usually where you start writing that. You can decide to click on a new file. This is an R script. You can decide to write a new R script. And then, so this is kind of a new page for you to start writing your code. And then you can always click on this Save Current Documents. If you click on this, you can name, just like the way you do it in MS Word, you can save it here and like write what that is for you. So, so going back to where we were. Yeah, so this is your source panel. This is where the software has an opportunity to yell at you if you make a mistake. This is where any of the new data frames or data that you put here, it appears here. If you want to import your data set, this is where you import your data set from. Okay, so you can always go here. So once you click on this, it allows you to look at your browser to pick out files that you need to use. It's just like what you do in Excel, where you need to grab the file from something. You can also do that here too. Okay. Okay, so without wasting much time, I'm just going to walk us through a particular project from start to finish. It looks like it's very scary, but really it's not. It's just a line of instructions about what the software should do for you. That's essentially what RStudio does. You tell it to do something for you and it does it. So libraries are kind of like what I explained before, where you keep your packages. So all the things that are inside of this are all packages in R. Like I told you, there are over 10,000 of them. If you need to find a package to do something in R, you can ask ChatGPT, you can type it in R. In Google, it will give you an answer of what to do. So it's really not a problem figuring out how to do something. So in this case, what I did here was to import this particular data set. So this is the name of data set, birth 14. The name really doesn't matter, but the idea is just that it's a data set that I imported. So it's somewhere in my documents or my downloads or my actual computer. So what I just needed to do was to retrieve it from there. So that's basically what this line is doing. That's inside of my folder, I have to look at downloads, and inside downloads, I should look for what this file is. And then I should grab that file and bring it back to R. That's essentially what this line is saying. So it's not, I mean, it's not, there's nothing very complicated about it. It's just tell R that you need to get this file from this place and then bring it here. So when you've been able to bring the data file here, one of the things you want to do is see what does this data look like. You might want to like check the structure of your data, like how many columns are there, how many data points are there. So that's one thing you can also do with this command. So you have functions that basically tell R what to do. Those are what functions are. They sort of tell R that, okay, this is what I want to do. So what this means is I want to see the structure of this data. That's essentially what that line means. And so when you run that, you can run that here. You can just run that. It shows you what the structure is. So it tells you there are 202 observations here. Okay, so this is kind of like the first line. I should run this first. Okay, so you run this first, and then sometimes you want to check what the structure is. So you run the next line. So yeah, so this is what it is. So you have 1,000 data sets, but in 12 columns. That's essentially what this is. You have 1,000 observations in 12 columns. So if you want to see what this looks like, you can actually click on, you can go here and see what this looks like. This is a place for you to view your data. So you can see there's 1, 2, 3, 4, 5, 6, 7, 8, whatever number of columns you have here. It's also going to be displayed there. And this is just showing you how many points you have on your data set. So that just gives you a simple way of looking at your, it gives you a simple way of looking at your, what's it called? It gives you a simple way of looking at your data set in terms of retrieving your code. Okay, so these are other, if you look at what's up here, it's just like other scripts that I've run in R or other data files that I've run in R. So you can always click from one to another just if you need to retrieve it easily. So now that we've seen what the structure of data is, it's obviously a very large data set, but let's assume we want to see a summary of those columns, like what do we have in those columns? You can always ask R for a summary. So that's what you are doing. So I want a summary of this data set. That's what that line is saying, essentially. So you get a summary. So now, typically you would have some sort of data dictionary that shows you what each of these things mean. But for the purpose of time, and because it's just a demonstration exercise, this refers to father's age, mother's age, whether or not the it's a birth data set. It's about like children that were born to a particular mother or a particular sets of mothers, like that, just pregnant women that had children. So it's just data concerning both the pregnant women and their offspring. That's kind of what the data set is about. So this is the father's age of the woman in question or any woman in question, the mother's age, whether or not the person was counted as mature or not based on a given age, like how many weeks the pregnancy was, like the visits, how many visits the person had, whether or not the pregnant woman gained weight or not, what's the weight of the child, whether or not the child is classified as having a low birth weight or not, what's the sex of the child, and all that kind of stuff, like habits, whether or not the mother smokes or not, and whether or not the mother is married or not. So that's just basically what these things mean. They're all variables or columns in our data set that contain information about the population that you're looking at. So in this place, you're looking at a summary of those things that are in this large data set. You can easily see what's the mean age of fathers. Like for each pregnant woman, what's the mean of their father's ages, or what's the mean of their mother's ages, or things like that. So that's kind of what this data set is. And then so you can see both for categorical and continuous variable what those things mean in this context. So it just gives you a glance of what you have in your data set. So it's possible that in such a large data set, it's possible that we have some missing data. So if you want to see if there's any missing data, this is a way to ask, is any of my data NA? NA is kind of like another way to describe missing data in R. So is any of my data missing? That's the instruction that is here. And then so you can see, so in places where you see true, it means that that particular place has any or is missing. So there's something missing. But in false, it means that it's not missing. So it's kind of like a question, is any of my data missing, or is any data set or any data points missing in this data set? If it says true, it means yes, it's missing. If it says false, it means no, it's not missing. Now, missing data is an entirely different question, a topic or subject to discuss as a whole in data science, and it's beyond the scope of this particular demonstration. So all this is just to show you that you can ask R to check your data set if there's something missing or not. Now, sometimes missing data can complicate your analysis or your visualization. So sometimes you might want to drop missing values, like you want to just remove all of those missing data from your value or from your data set as a whole. So this is a way to do it that, so this is an object. These are what we call objects. It's kind of making things easier for you to handle in R. So in this object, what I want to do now is that this, my data set, I want to omit the missing values from that data set. So when I click on this and give this instruction, it drops, what it does is that it drops the missing data. That's what it does. So now, how do I know that it has actually done what I told it to do? You can actually check the summary again and see if there's like, okay, so if the summary is also one of the ways you can look at whether or not you have missing data. Because if you scroll up here, sorry, this is a lot, but if you scroll up here, you can see NA values here in the summary that I ran before. So this was the summary I ran before. The first one I ran on the unedited data sets. I can see NAs. It means that in this column, 520 points are missing. So, but then in the new one that I ran, after I've told R to remove the missing data, you can see that that is no longer there, meaning that R has actually done what I told it to do. So in this new data set that has been created within my environment, there are no missing data points. It has dropped all of the missing data points. And so now, of course, you can also check the structure of your data. Now, if you check the structure, you see that this is 202 observations from the 1,000 we started with. So clearly, there were so many missing data points in the original data that we started with. So like you've lost almost 80% of your data by dropping missing data points. Now, that can be problematic depending on what you are doing, but because this is a demonstration, it's not going to be a problem for us. That's like another subject entirely. It's a much difficult scope or concept. So we've been able to drop that. This is the new data set. We have 202 observations with 12 columns. Now, so what if I don't want the 202 observations? What if I just want to work with a small portion of those observations? I just want to slice it and take a small part of that and work with that. What does that look like for me? Okay, so sorry, I'm just going to run through this now. So this is what it does. You slice the sample essentially. I want 175 from my data set, and that's what you've told R to do. That I just want to take from the 202. I just want you to slice 175 rows and just use that for me. And then so now that I can also look at that here, so let's view what has happened now. You can see now that there's 175. There's still 12 columns, but it's 175 observations because I've been able to slice that. So you're able to do all of this in the same environment in R. That's one of the good things about R. So let's say I don't want this column. I'm not interested in whether or not the mother is a smoker or not a smoker. Like, so I just want to drop this particular column. I'm not interested in figuring out whether the mother is a smoker or not. I can tell how to drop the column. So I can count it. So this is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So I want to drop this 11th column. And that's essentially what I told R to do here, that please drop this column, this 11th column from me. And then when you do that, it drops it. And then you can look at the new, what's the new data set now that has been dropped. You see that that's no longer there. So you can tell that's to R to do. So if you just want to visualize something, you want to explore your data or see stuff, you can use base R to do plots. You can just say what's the distribution or how does the father's age look like. You can look at what the weight is, like in terms of who gained weight or who didn't gain weight. You can look at that. You can just see a distribution of that. And you can also do a bar plot, like to see for categorical variables. That's variables that are arranged in categories. You can see from that, like using base R, what the distribution is like with respect, do we have more males or females? Like that's a question you might be interested in answering. So you can also explore that with base R. Now, this is very complicated because a lot of times like these are just standard. There's no way to walk around if I want to change the color, for instance, if I want to change the height of the bar, if I want to change anything in R. So that's why ggplot is such a or ggplot2, depending on which you want to use, is such a useful thing. It's kind of like another package in R that helps you to make certain types of adjustment to my data, to customize it so that I like what I see. So of course you have to call the library and tell the library that, okay, I want this book known as ggplot2. I want to use it for something. In this case, we want to use it to visualize our data. So let's assume that I'm interested in creating a bar plot for the same sex that we did, but this time around, I want to use it in ggplot. I can use that to, what's it called? I can use that to show the different categories. So, this is slightly different from what I did now because it's much nicer to look at. And I can say, okay, this is females and this is males. Like in terms of the offspring, like the child that was given birth to by a particular mother, by the mothers as a whole, what proportion of them were females, what proportion of them were males. This is the way I can do that. Now, let's say I don't like the way this looks, and I'm just like, oh, I don't particularly like the way this is. I want to color it by sex. I don't want them to have the same colors. You can also do that in R by telling R what you want. So I want this to be by sex. That's essentially what fill means. You want to color it based on the sex. And of course, you can also color it based on the sex. And then you get different colors based on how that looks like. So what if I want to change the background? I don't like how this looks like. I want it to be plain. I want it to be minimal. You can add a theme classic and tell R that I want that to be changed. So everything else is still the same. You're just telling R that I want you to change. Yeah, and so you have something white as the background. So it's nicer, it's neater for you to use. Okay, so let's assume that you're also interested in viewing. So we're almost at the end. So let's assume that you're interested in viewing a content. So these are categorical variables because they're in categories. But let's assume you want to view something that is not in categories, something that is continuous like age or weight. So you can also do that in R. You can simply give an instruction to R to do that. Okay, we can tell R that you're interested in doing that. Now there's a problem. We don't see any output here. So what exactly is wrong? It has given you a couple of warning messages. So this is where I mean that the software is basically yelling at you and telling you that, hey, you did something that I don't like, so fix it. And so, and sometimes you might be confused as to like, I don't exactly know what I did wrong. So what can I do about that? One option is that I could ask ChatGPT. So you can copy the error message from here and paste it in ChatGPT and you get a response. And ChatGPT can also tell you how to correct your code to get the right answer. And that was what ChatGPT did for me here. So ChatGPT basically took the code that I put in and told me how to adjust it to get what I want. So now that I've adjusted that, voila, I get my results. I can now get a histogram of age. But this is boring. That's why I want colors. I want to color the different bars in R, like in this plot. I just want something nicer. And I don't like these grid lines. It's disturbing me. I want something nicer. I can also tell R that I want something like that. And so I want a minimal thing. That's essentially what that does. And then you also tell R that you want to color it based on the bars. And you can also run that in R and see that, well, I want the lowest one to be light blue. And then I want the highest one to be dark blue. That's basically what you've told R to do. And then R has done the same thing for you. And you've done a minimal theme and all that. So you easily can see how you can adjust that now. So essentially, the purpose is just showing you that there are so many things that R is capable of in terms of visualization of data, handling data, and just trying to interpret data as a whole. One thing I did not mention was that these instructions that are attached here, you can also attach them by just using the pound key or the hash. So if I remove the hash, there's going to be an error. If I run this, you can already see that R is yelling at me here. Now, if I want to comment, I want to comment to make it easier for someone to understand what I've done. You can just use that to add code and commenting. So this is a quick demonstration. Now, all this code that I've done, supposing I want to submit it as part of an assignment, what do I do to export all of this code and submit it? A simple way to do that is to just use a compile report. It's here in your RStudio source file. So you can compile your report, and then you can choose the form you want it to be in: PDF, MS Word, HTML. Remember that this is what we are compiling. This is where we want to compile it. And then we can compile that. Now, sometimes you have an error. It's possible that you have an error. That happens very often. And that could be because like you made a mistake somewhere in the... Now, if you remember where we made a mistake with ChatGPT here, if you leave the mistake here, it's not going to run when you're compiling it. So you have to close that mistake so you can allow it to run. So because the point is, it's going to run everything from start to finish. So if there's any problem anywhere in the middle of your code, it's going to give you a problem, like it's going to give you this error. So now that we've sort of closed, so once you put that hash sign, it ignores that part and it doesn't run that. So now that we've ignored where the error is, let's see if we can run it in HTML. Hopefully we can. Just like wrapping up in a few minutes here. And oh yeah, precisely. So this is exactly what it looks like. So this is your HTML. It includes all the codes that you ran, all of the results that you got. It shows both your plot, it shows all your response, all the things you got as like values and responses and like your output. It runs everything and compiles everything for you in one place. Yep. And so that's what it does. And so you can easily see all of what you've done from start to finish. So that's one way you can export some of the things there. You can also do it in, like as a Word document and all that. So, but I wouldn't go into that because of time, but it's really a nice thing to do. Now, the point is you're not you can actually learn to do the same thing I've done in less than a week. It's really not, it's just about understanding what everything is here. There's no problem with respect to like, oh, if I run into a problem, what do I do? You will run into problems. It is 100% guaranteed that you will have an error in your code, like when you start running it. And it can be frustrating when you start, but that's essentially how you learn how to use R. Like you make a mistake and you figure out why I made that mistake and you try not to make that mistake again. And so that was where I mentioned that slide about debugging and what you need to do if you run into a problem. So hopefully this provides some sort of explanation as to how to use R. If you don't like black, if you are in the category of people that don't like black for your background, you can go to your tools, go to your global options, go to your appearance, and be like, I don't like this black. I don't know why this particular presenter is using black. I'm not the kind of person that uses black. I want to change everything to white so I can see it easily. You can apply that and everything changes back to white. Now, once you start using black, I'm very sure you see how different it looks on your eyes once you change it back to white, because we've been using black like since the beginning of this presentation, but please, let's go back to black. Sometimes I can also adjust the font. I want my font to be bigger. I want it to be smaller. You can make all those adjustments in this area, and then you can apply that and sort of use that. So it's now, if you see this is a much lighter, it's black, but it's not as sharp as the black I used before. So it's your preference really matters. Whatever works for you, really, that's basically how it is. So I hope this provides some sort of like explanation of using R. I know the time was short, but my email is going to be attached. So if you run into problems, in fact, I would want you to replicate the same thing that was done. I would be able to provide the codes for you to do this and provide the data set, and then you can do amazing stuff and then share the things you've been able to do with me. If you run into problems, let me know. If you need to know how to use ChatGPT to figure out bugs in your codes, like that's something I'm willing to provide guidance on. I would say for maybe the next 2 weeks, and then I have to jump onto something else. But I hope this provides some explanation of how R itself can be such an easy thing to use. I'm not saying you would always figure the answer out. Sometimes you might run into problems figuring the code, and sometimes you might be there for like 2-3 hours. And the only problem could be that you did not, it might just be an issue of you not putting a comma here. And let's say that's the only problem, that let's assume that's what you make a mistake with, and you keep on running the code and it keeps on yelling at you that there's an unexpected symbol. Now, so you have to be like, why is there an unexpected symbol? What's going on? You can be on this particular, something as simple as this can take you 1-2 hours to figure out. So that's an example of something you can use ChatGPT to do. So, but you figure out that once you attach the right thing and then you run it, it feels like magic, and all of a sudden, oh, sorry, you're supposed to run everything together. And then so you get the answer. So that's essentially what you're going to be doing for like a large portion of the time, and figuring out how to do something that in R. And so hopefully this provides some explanation, and I really wish we had more time, but I'm grateful for you listening. And so yeah, I've opened the floor for questions now. I'll probably still leave this on because you might have some questions about this. Yeah, so thank you for listening, and yeah, I'm ready to take questions.
Okay, yeah. Thank you very much, Bamgboye, for taking us through the interesting and technical section. Yeah, when you said how is majorly to make life easier, I was like, okay, yeah, I'm ready for this section. But after seeing the How Studio window, oh my God, it's so scary. But like you said ChatGPT is there for us, then yeah, we can check through and ask questions, and yeah, maybe we can go through the how and everything. So if you have any question, you can put it in the chat box or simply signify with an emoji. Yeah, I already saw a question in the question and answer segment.
Okay.
The person wrote, please, what is the meaning of like a code? I don't know.
Okay, so that's like a pipe command. That's a pipe command. So piping is kind of it's a way to make your life easier. Instead of you writing a code like multiple times, it's kind of a way for you to compress everything in one, like do multiple steps in one line of code. Okay, so let me go back to, I think, now, so this pipe command here, yeah, I know it's used a lot of times. So what it's trying to tell you is that in this birth data set, I want to omit this in this birth data set. That's what I want to do. So instead of me now, it's kind of like saying do 2 things or do 3 things or do 4 things at once. That's kind of what it does. So it's like I would say it's kind of like a conjunction. If you're looking at it in terms of grammar, it's like a conjunction that I want to do one thing and then another thing and then another thing and then another thing all together in the same line of code. That's essentially what it does. hopefully that like helps the person that asks the question. So
okay, yeah, thank you very much for the answer. Then we have another question, and it says, when you save your code in HTML, how would you be able to rerun it?
Oh, okay. So essentially, that was something I did not show that I should have also shown, is that or showed rather. So like what I was going to say was that you can name this data set. So let's assume, since we can all see my screen, I can copy all the code here because I've already named this. I would say I'm biased. Let's say I want to start a new project now. And let's say I run all this code from start to finish and all that kind of stuff. And so I run all the code in this particular place and all of that. So let's say this is a new, now you can see this is untitled for, it's untitled, I don't have a name for it. So let's say I want to save it. I can save it as demo, let's say demo, whatever. I can say demo for Science Cafe. Okay. So I can save it as demo Science Cafe, and then I save this. So it's going to be saved as a file it's going to be saved as an R file in my document, just like the way you save a Word document. So when I open that in my own, let's see. So hopefully we see, okay, can you see this page? Can you see my document as a whole? Is that something you can see? Or you're still seeing the
Windows, the House Studio window?
Is what you can see. Okay, so I think the problem is I didn't share. So since this is debugging, I think I have quite some time to show us. Okay, so what I'm going to do is I'm going to share my window. Okay, just a minute, please. So please let me know if you can see. Okay, please let me know if you can see this, like my Word document. Sorry,
Yes,
my document, my file folder. Sorry. So you can see demo. We can see this demo Science Cafe that I just saved now. So if I click on it, let's assume I share it with someone. If I click on it, this is what it does. It opens this page for me in RStudio. So it's kind of like what you do when someone sends you an Excel file and you download it, or a Word document and you download it and you open it. It's going to automatically take you to MS Word or take you to Microsoft Excel or something and open it. So it's the same thing with RStudio too. So you can share that document to us, like maybe if you want to submit an assignment or something, you can submit that and you share that. Or if you want to copy and paste, some of us like copying and pasting is a very nice thing for us to do. So I can copy my, the way I did it as an HTML, I can copy that, paste it in a Word document. Let's see, I should be able to do that here. So I think that was the Bambi one. Okay, so this Bambi one, now let's assume that I want to compile it. So I can compile it as an HTML. You know, you can also choose Word in that same place I showed you. So let's assume that I copied it in a, so it appears as an HTML now. Everything is basically here. So I can just do a Control A and a Control C, and then I go to somewhere like some Word document somewhere, and I what's it called? And I just decide that I want to copy it exactly the same way. And it brings everything back. I can copy it back here. I can convert this to a PDF. So there's just like, and of course, Bambi, whatever. So it's exactly, this is another way for you to do it. Like you want all of your code. But the thing with this is this shows you both the code and the result. So it's likely to be something you may want to show for maybe like if you are submitting an assignment or something. So it just depends on how you want your output to look like. Okay, so I hope this provides like some explanation for the person that asked the question. Does that, I hope it does.
Yeah, she said it does. So yeah.
And there's something else too you need to know. There are usually multiple ways of doing things in R. Like I'm very sure like for the same thing I've done here, there could be like 3 or 4 other ways of doing the exact same thing. So that's one thing you should be open to with using R. Like there's a lot of times there isn't one way of doing something, unless if you get into very complicated things, like maybe you are trying to do some maybe like survival random forest and like machine learning or something. Like once you start getting to a bit more technical things, there are fewer options to do those things. But for many of the general data manipulation that you do in R, you get, what's it called? You get multiple, there are multiple ways for you to do that same thing in R. So like, it's just a function of what someone's preference is for that. There's a question about, so R is basically manipulating data. Well, in many ways, yes. In many ways, it also does statistical analysis, which I did not show here. This is probably an example of what that could look like in terms of, this is another project I was working in, for instance. So I'll probably just run all of it now and just show you what that looks like in terms of, yeah. So this is an example of something else. So I did an analysis and all that, and this is the result. I just wanted to produce a table that I could submit. The table is the only thing I'm submitting. I'm not submitting all of the code. I don't need to submit the code. It's just the table I'm submitting. So while R is also a very nice thing for like manipulating data, it's also very useful for statistical analysis and then machine learning. This is like a machine learning project. So you can easily use it for machine learning too. So it's really, I don't think it's a nice way to explain it, but it does a lot of things. That's just the summary. Like it helps you manipulate data, visualize data, run statistical analysis, do some machine learning work and all that kind of stuff. So that's also very nice to do with R. Please, more questions. I am excited to take questions.
Yeah. So thank you very much. We have one hand raised, and
oh,
that
yes,
is
yes, please. So,
yeah.
Thank you, Bamgboye, for such a lovely presentation. I would just like to ask, do you have any tips you would like to share on how to use the help function in R?
Okay, yes. Thank you so much for that question. Now, that's something I run into problems with every now and then, because sometimes I need to do something in R and I don't know how to do it, which is something that is quite, what's it called? It's quite common. It's something you would face. You know, like I've said it before, that expect to run into problems in R, that it would happen. It's 100% sure it's going to happen. So sometimes what I do is like, you can click the help function. You know, there's this help tab here at the bottom right. You can click on the help tab and say, let's say I want to do a histogram and I don't know how to do a histogram. I can just type histogram in that place and let it give me a question. Oh, sorry, and let it provide an answer for me. So it gives me the documentation of what to do. And then, so this is how to use the histogram function. It's like, okay, this is what I do. I have to write a histogram first. And then I have to write what I want. X is the object I want to plot a histogram for. And then it gives me a lot of other advice and suggestions and tells me what each of those things mean. So all these arguments that are inside here, like what does aspect mean? I can always go down here and look at it and be like, okay, what does aspect mean? And then I can check what aspect means and all that kind of stuff down here. And so it gives me all that. It's like a documentation. It's like a how-to guide of how to use it. And you can also run an example here. If you click on run examples, what it does, it shows you, it gives you an example of how to do what you want to do. So yeah, it's saying that you need stats. Stats is a package. Sometimes it's in base R, sometimes it's not, but it's usually in base R. So if it's in base R, you don't need to install that again. So this is basically one way I can do it. This is another way I can do it. If I want a density plot, this is how to do it. But sometimes I might just be like, oh, I don't know what to do here. Like this is very confusing for me. So can you see the ChatGPT space? I'm hoping you can see that.
Yes, we can.
Okay, so you can type here, how do I make a histogram plot? I can't spell anymore in R. So if you click on that, how do I make a histogram plot in R? It's, of course, this is quite slow because I'm running multiple software at the same time. But usually what it does is that it gives you some sort of results. Let me see if I can, this is, okay. It's creating a response bar. This is taking quite some time. But the point is, it's going to give you quite some, it's going to be very, okay, sorry about that. So how do I make histogram plots in R? ChatGPT is telling you how to do it. So this is the package you need to do. This is what is recommending, which we've already used before in the example. So you have to load it. You have to, what's it called? It gives you the argument. This is essentially what you're supposed to do for you to get that to run. And then it's also going to tell you that, okay, I need to replace data with like what my data actually is. So in this case, it's like the birth, 14 new, whatever, like that's it. What's the variable I want to plot for the histogram? Let's say is weight. I have to tell you that weight is what I want, and then all that kind of stuff. So you can use ggplot too. You can also use base R which is like histogram and all that, which is kind of the same thing you saw in the help function. So this is one other way to do it. You could also type online in like your Google, like how do I plot a histogram in R? So, but it's a good idea to always look at the help function and use that to check what I need to do. Another way to do that is to also use a question mark. So if I want to do a histogram, you can use a question mark and then say histogram. It's like, how do I do a histogram in R? It gives you the same thing as what it is here. It shows you what to do. So you can, if you need to do something in R, you need a function and all that, you can put a question mark before it, and then it displays to you what you need to do in R. Like, this is how you should use that function. But of course, like I said, there are always multiple ways to do things. So ggplot is another way of doing it. So if I'm like, oh, I want to use ggplot, like you can also type ggplot here or ggplot2 and be like, oh, what does ggplot do? It tells you what a description of what ggplot is. It tells you the people. So ggplot is the package. So these were the people that came up with it. So you can also come up with your own. And then you can look at the whole like description and all that documentation and description of what this actually does. It also provides you some extra links if you want to look at what the people did in the original package and all that. So anyone essentially can write a package. So that's basically how to use the help function. Hope that's helpful. I know you're asking about help. I hope the comments about help was helpful.
Yes, it was. Thanks very much.
Okay.
As to why it's called R, it's named after the creators like Ross and Robert, like at the University of Auckland, New Zealand. Those are the people that came up with R. And it's kind of like they built upon a previous language known as S. So that it sort of like utilizes S essentially, like and all that. So there have been so many programming languages before. So R is kind of like did I say it's a modification of S? And so the people that came up with how to use the software as a whole, they are Ross and Robert, like at in New Zealand. So I'm assuming that that's why it came up. I've read it somewhere before that that's how it came about. Hopefully I'm correct as to why that is. Yep.
Okay, yeah. Thank you very much, Bamgboye, for the detailed and the technical presentation. I really enjoyed myself, although it's so much to take all of it in tonight, yeah. But then I enjoyed the section. So on this note, I would like to say once again, thank you very much, our guest speakers for tonight, Dr. Jaiyesimi and Bamgboye, and everyone present here tonight. And please don't forget that the anniversary event still continues tomorrow, Saturday, and starting at 7:00 p.m. So yeah, on this note, we have come to the end of the section tonight. And yeah, you do have a wonderful night's rest, everyone. And for those that are still in the morning, do have a wonderful day ahead. Thank you, everyone. Thank you, our presenters. And thank you for honoring our invite. Thank you all.